So, dear colleagues, we would like to start. Please take a seat. We want to be right on time. So, on behalf of the ESPID board, the ESPID committees and the organizational team, and also on behalf of Susanna, who will chair the next session, and my co-chair, Liat Ashkenazi Ofnung from Israel. My name is Tobias Tenbaum from Berlin, Germany. We welcome you officially to ESPID 2026 in Bologna. We are all happy that you are here attending this meeting. And as far as I heard the rumors, it has an extraordinarily high number of participants, thanks to Susanna for this, but we'll get to this later on, of course. Now we start the first session on the topic of climate change, vectors and change in geography, infectious diseases, not only pediatric infectious diseases, but also in general. And we have three experts that touch that topic from different angles. As you know, climate change affects us in every situation of our daily life, and that's why the International Scientific Committee put some efforts to carefully look from different perspectives on this phenomenon and how it touches infectious diseases. So I'm quite happy to introduce the first speaker. And what can be more perfect than starting with a person from Bologna. So it's Dr. Romeo Bellini, and he's a medical entomologist and the former director of the Medical and Veterinary Entomology Department of Centro Agricoltura Ambiente de Nicolo in Italy. And he has several experience in mosquito ecology and vector-borne disease. And I'm very happy that he starts off our science slam on vector-borne diseases, and the floor is yours.
Thank you very much. Welcome.
On a side note, if you have comments, please also use the app to post questions. You can also, of course, after the talk, go to the microphones and ask questions, but please also use the apps and switch up otherwise the sound of your phone. Sorry. Now, your applause.
Thank you. So when I got this invitation, I realized that One Health principle is working, because after a long career, it's the first time that I've been invited to such an important meeting. Usually we have medical entomology meeting, small meeting. In the Society of Vector Ecology we have in Europe, we have no more than 200 people. One Health is the future, and already here. I have nothing to disclose. I think that it was mandatory for the organization. So, talking about climate change, I prefer to start dealing with the term Anthropocene, meaning that our species has achieved an impact, huge impact in terms of ecology of the planet. Climate change is just a small piece of the picture. Deforestation, urbanization, irrigation, poverty, trade, and travel all has an effect on vector-borne disease. And this is not always easy, not always possible to discriminate between the complexity of the system. WHO, of course, consider vector distribution, vector ecology, and vector-borne disease as a part of the puzzle, together with many other health risks that compose the climate change scenario. When we talk about the European region, I like to just put some name. Considering mosquito-borne disease, we have dengue fever and chikungunya fever, which are not endemic, but are continually introduced through travelers, infected travelers. Malaria used to be endemic, is mostly eradicated. We have some fauces of malaria Plasmodium vivax perhaps in Greece, small things. West Nile fever is becoming endemic in several areas. Sandfly-borne disease, leishmaniasis, you will know. Tick-Borne, I saw there is a presentation after my introduction, so I will not go inside the Tick-Borne disease. In terms of global annual incidence of mosquito-borne disease, we have numbers which are not precise because, especially in Africa, statistics are not reliable enough. By the way, we have an estimation about malaria is the most spread disease with more than 200 million cases. Dengue is rising, more than 1 million cases. Lymphatic filariasis, chikungunya, Zika virus, yellow fever, despite the vaccine very effective, still have a huge impact. Japanese encephalitis and West Nile virus. Doesn't like the next one. So the most important thing to consider this is that arthropods, the vector of disease, are heterothermic, meaning that they cannot regulate the body temperature, and therefore they are exposed to the environment. This is very important because it's the main reason why in temperate areas such as Bologna, for example, we still have winter time without mosquitoes or without sandfly for 6 months because they cannot regulate the temperature. And this is a very important factor, of course, influenced by climate change. When we talk about climate change, vector-borne disease drivers, we will focus a little bit on the time of development of the vector, the cycle of development, the vector ecology. Climate change affects the vector geographic distribution and may open the door to the spread of invasive species. We will see some example. It's changing the risk seasonality. It may be longer the risk season, and it affects the pathogen development cycle and also the human behavior. We have evidence in Africa where humans, because of the hot season, tend to sleep outside, and sleeping outside, you are more exposed to have a disease. This is a simple development cycle of mosquito. Mosquito, of course, as you know, the female requires blood meal to develop eggs. All these phases related with temperature, each species has a specific requirement in terms of ecology. And when the mosquito bites an infected host, if it's competent, it may acquire the pathogen, and after a few days, depending again from the temperature, it may become infected and be able to infect a new host with the next blood meal. All these phases are regulated by temperature and, in the case of mosquito, by water availability. For example, the gonotrophic cycle, meaning the time required from the blood meal to the egg maturation and the egg oviposition, is strongly dependent from temperature. It may go, in this case, from 5 days at 20 degrees to 2 days at 34 degrees. And this makes a huge impact on the epidemiological capacity of our species. It is important to consider that we have a maximum threshold, which is different for each physiological phase. Above this maximum threshold, this optimal threshold, we have a decrease. So this is a question that has been already considered by modelers that like to predict different scenarios considering the temperature, the optimal temperature. Situation may be more complex. In this case, for example, which is a 3-species vector of West Nile. Likely we don't have this species We don't have all these species in Europe. It is related to the United States. There is an interesting point, which is a counteracting question, meaning that when the temperature increase, the survival of infected female decrease. So in this case, it may be counterproductive for the system to have an increase in the temperature. Another piece of the job is the extrinsic cycle and temperature. And this is the picture considering West Nile and Culex pipiens. Culex pipiens is the most important vector of West Nile in Europe. You see that at 20 degree temperature, the pathogen cannot achieve the salivary gland, so it cannot be able to introduce an epidemic, while at 30 degree, in few days, such as 2, 3, 4 days, the pathogen can go through the mesenteron, the Malpighian tubule, and achieve the salivary gland and be ready to be transmitted to the next host with the blood meal. All these components shape the epidemiological equation, which is this is a simplified version of the epidemiological equation made by Fang and Rising back in the 80s. We see that I put in red the parameters which are influenced by temperature. Not all of them are influenced by temperature, but vector competence, for example, which is the capacity of the species, the efficiency of the species to transmit a pathogen is related with the temperature. The survival of the female is related to the temperature, and the duration of the extrinsic cycle is related with the temperature. While the number of blood meal or the feeding range, so the activity related with birds, mammalian, or other hosts, is not influenced by temperature. Just to communicate to you that the scenario is quite complex and need to be considered case by case. I'd like to show you this picture, which includes the variation in the malaria case incidence and the number of deaths due to dengue in the last 20 years, not the last 20 years, sorry, in 20 years from 93 to 2013. These are official data by WHO region. It is interesting to see that we have a strong decrease in the number of malaria cases all over the region by 50, 60, 70% decrease. And this is related with the Anopheles control and the malaria control system that have been developing, especially the long-lasting treated net and the internal residual spraying, while in the same region we have a huge increase in the number of dengue deaths by 6 times, 3 times, 4 times, 6 times. So this I consider this a nice picture showing that we cannot establish general rule. We need to check case by case the situation. Considering modeling and the estimation for the end of the century malaria risk in Africa, they establish that there is an expectation of a reduced incidence of malaria in several countries in the sub-Saharan Africa and in South Africa, while we have other regions where it is expected an increase in the malaria risk. And this is also a matter of altitude, mountain area, water availability, and other factors. Leishmania in Europe also is complicated. We expect some area with an increasing in the suitability of Leishmania, while in other areas we expect to see a reduced incidence. Also considering modeling, I'm not an enthusiastic supporter of the modeling, but I think that the modeling can have some impact in making us thinking and seeing things in a different way. This is nice because they compare the observed climatic data from the beginning of the 19th century until now, and the simulation without temperature increase. And they were able to show that the temperature is playing a major role in the spread of West Nile in Europe. Out of the temperature, there is the extreme event increase, such as flooding. In general, I saw there is a conviction that the flooding, after the flooding, we have an extreme development of mosquitoes and an increase in related disease. This is not always true. For example, this case where I was involved directly back to 2014 during the flood of the Sava River in Serbia, Croatia, and Bosnia and Herzegovina during June, so in the perfect timing for mosquito development. We were called by WHO because of the risk of a West Nile epidemic. We didn't see any increase in case, both in 2014 and in 2015. So there was no evidence of such an impact. And also it is interesting to show that our colleague in the United States, as you know, is exposed frequently to hurricane that may have an impact on mosquito and related disease. They have a number of arbovirus listed here. And the case where they see some increase can be related to hurricane is very limited, very limited. I like also to mention the exotic species spread due to climate change. We have a huge impact of new species, algae, fishes, crustaceans, mollusks in the Mediterranean Sea, which are related with the temperature of the water. In the case of our mosquito, Asian tiger mosquito, Aedes albopictus, temperature is not important in the spread, because the most important thing is the fact that the species has been introduced by used tire, trade. Millions of used tire are stocked outside. They collect water when it's raining. The mosquito can develop in this water and can be spread by transportation. With the presence of this mosquito in Europe, we have now some limited outbreak, local transmission. In the last 15 years, we have also one outbreak in 2007 in this region, and we have also evidence of outbreaks due to Aedes albopictus in southern France. And considering dengue, we have evidence of outbreaks due to the introduction of the virus through infected travelers in northern Italy, southern France, and Spain. As a vector ecology and medical entomology, we are now very worried about Aedes aegypti introduction. Aedes aegypti is the most important vector of dengue, I would say chikungunya partially, and yellow fever in the world. It's a long story. It was present until the Second World War in several parts of Europe, and it disappeared, nobody knows why, the reason. It is now present in the East Black Sea coast, Turkey, Georgia, and some part of Russia. And due to the climate change, we expect to see the species in the Mediterranean basin, and we are strongly working in order to prevent, because if this species will be introduced, will change drastically all the public health scenario. So, as a take-home message, I would like to say that available findings indicate that the net effect of climate change on vector-borne disease transmission may be context-dependent, reflecting competing influence of physiological impairment and adaptive or tolerance responses in exposed vector pathogen system. And with this, I thank you for the attention, and I'll be here to answer possibly to your question. Thank you very much for your attention.
Thanks, Romeo. We'll continue and take the questions at the end of the whole session. So I hand over to Liat to introduce our next speaker.
So thank you, Dr. Bellini, and we move on to our second speaker, which is Dr. Pablo Martinez de Salazar, who will talk about climate-sensitive infectious diseases, epidemiology, prediction, and public health response. Dr. Martinez de Salazar is a physician and ID researcher, currently a project leader at the Swiss Tropical and Public Health Institute, affiliated with the Harvard Hahn School of Public Health. His work focuses on climate-sensitive infectious diseases and the translation of surveillance data into public health action. Please join me in welcoming Dr. Martinez de Salazar.
Hello, thank you very much. It's a pleasure and an honor to be here. I have to declare that I have nothing to disclose. So I'm a clinician by training, but I work on mathematical modeling and complex data analysis. So if we would like to answer the question how climate change impacts the risk of infectious diseases, the perfect experiment will be to have two copies of the world. In one, we will have climate change. In the other one, we have stable climate, and then we will measure disease outcomes. Obviously, this is not possible. We have to rely on observations, and with observation, we have all the problems, classical problems that we have with causal inference, which is association is not causation, and you have simultaneous changes that are happening, such as how we use land, how we move, how good our surveillance systems are, that are confounding what we are measuring. So the challenge here is how we infer causality, given that the only thing we have is observations, or as we can call them, natural experiments. So within infectious diseases, vector-borne diseases are very interesting because they rely on complex ecological dynamics, and many of the pathways are sensitive to climate change. Among vector-borne diseases, tick-borne diseases are the most important ones in Europe from a public health perspective, and children are particularly vulnerable to ticks because they spend a lot of time outdoors. They have naive immune systems, but also once the ticks are attached, it's very likely that they remain unnoticed for longer periods than in adults. And this is relevant because, for example, for transmission of tick-borne encephalitis or Crimean Congo hemorrhagic fever, we might need just only a couple of minutes, while if for Lyme borreliosis, usually we need at least 1.5 days or 2 days. So I will present my work on Lyme disease and tick-borne encephalitis in Switzerland as a case study. Switzerland is among the countries with the highest susceptibility to climate change because of geographic location. Indeed, since the pre-industrial era, Switzerland has increased by 2.9 degrees compared to the global average of 1.3. Also, Lyme disease and tick-borne encephalitis are the most important tick-borne diseases in Europe. Lyme disease because it has a high incidence and we can prevent the complications from dissemination. And tick-borne encephalitis is kind of the opposite. It's a virus that it can be prevented by vaccine is rare, but it can become severe, particularly because the virus is neuroinvasive. So in children, it's not so much about mortality, but about long-term neurodevelopmental or cognitive effects. So in Switzerland, less than 10 years ago, the risk of acquiring tick-borne encephalitis was relatively localized in specific spots, while nowadays, if you go there, you can get the disease overall. Similarly, some colleagues in Zurich saw that the number of cases of children attending the emergency clinic with Lyme borreliosis has almost doubled in less than 10 years and represent 1 in every 700 pediatric emergency cases. So the first thing that we did was to try to figure out what is the evidence that we have. So we know that there's an association between climatic trends and tick-borne diseases, and this has been very robustly shown in different countries in Europe, but also in North America. We also know from observational data and experiments that certain climatic conditions affect ticks. For example, we know that milder winters are associated with increased survival of the ticks. We know that there has been an increased distribution in terms of higher in mountains and northern latitude in Europe associated with warmer temperatures. But on the other hand, we know that drier and hotter environments reduce tick activity. So what we did, we built what is called a causal framework, where we try to figure out what are the pathways that link climate to the risk of infections. And as you can see, we identify 4 different domains: ticks, animals, humans, and pathogens. But the truth is that, as I said before, it's not only climate what is changing, and a good example is sports. In Switzerland, in the last 40 years, there have been an increase in number of people who are doing sports and the frequency that they do it. And number 1 sport in Switzerland is hiking. So roughly 60% of the population regularly hike, and usually in natural places where they can be bitten by a tick. On the other hand, in Europe and in many other places in the world, people have moved from rural areas to the cities, and that means that they have stopped using the land for primary activities such as agriculture and farming. So what we see in Europe, and particularly here in Switzerland, we see a natural reforestation process that has been taking for decades, and that involves that we have that the animals and the ticks have recovered habitats that they didn't have before. Also, we have seen in Europe that there has been an increase in the density of big animals, for example, deers, which are a major host for tick-borne diseases. And this is partly explained of this recovering of the habitat, but also because obviously we hunt less if we are in the cities. So, and this increased incidence actually overlaps with the areas where we have a higher incidence of Lyme borreliosis and tick-borne encephalitis. So when we build out this conceptual framework, we really need to recognize and acknowledge that there are other processes that are not climate that are simultaneously changing, for example, how we use land, how the land reforests, on how human behaves, how much time we spend outdoors, that are independent of climate. And we have to recognize them to really ensure that they are not confounding. So the second thing that we did was to gather data from Lyme borreliosis and tick-borne encephalitis for almost 20 years. The data from the Lyme disease comes from a surveillance network in Switzerland, which roughly represents 2% of the catchment population. And the tick-borne encephalitis, it's a reportable disease, so this probably represents symptomatic cases that reach the health access, so they were serious enough, and they were confirmed. So even if this doesn't represent the total number of cases in Switzerland, the trends, how they change every season and how they change every year, it's actually a good representation. We also obviously cannot monitor ticks all over the country, but what we can do is check, we know the weather in every place, every minute in Switzerland for the case study period. So what we did, we build up, we estimate the weather in small squares all over the country and figure out when were the optimal weather conditions, which involves temperature and humidity, that really allow for tick activity. And zooming up all this massive amount of data, we can build up a time series over the case study which represent how active the ticks are depending on climate. Also, we don't know how people move and how much they do all the time in every place, but actually here we approach this using something that has been previously successful in other infectious diseases, which relies on the assumption that what people search in Google is actually a good proxy of what people do afterwards. So we use search terms such as hike in the different languages in Switzerland, and we build a time series every month. And this time series aims to represent how much people goes outdoor and how much people are active in places where there are ticks. And actually, we were able to validate this approach by using mobility data that was available during 2020. So we saw a very strong correlation and a very strong prediction from this Google search data in when the people were going to natural park and forest areas. So we have what we want to predict, which are the cases. We have a proxy for tick activity. We have a proxy for human activity, but we are missing the animal host based on our framework, the animal host and the habitat. And this is the part that I like most. So a couple of years ago, some researchers published this interesting study where they have been sampling ticks in the same spots for many years. And they saw that there was a very strong association between the tick density and masting. Masting is the production of trees, of seeds by trees. And masting has two characteristics. One is that it does not happen every year, so trees don't produce fruits every year. They will produce fruits 1 year, and then they will rest 1 or 2, 3 years, and then climate will show a signal like increasing warming temperatures, and then the trees will be able to mast again. The second feature is that masting is a synchronous effect. So actually, not every tree masts differently, but all the trees in the same forest mast at the same time. And actually, many forests at the same time mast at the same time. And indeed, in Switzerland, we observe national level masting effects. So you can see the trees, this is a beech tree, which is particularly relevant, masting at the same time. So what these researchers found is that they could predict the tick density by looking at the masting events 2 years before. And what is the explanation for that? So seeds are the major source of food for rodents and many other animals. And rodents, in turn, are the major source of food for ticks. So what happens is that you have a big masting event in a forest, then the rodents, the mice and the rats can feed on it, their population explodes, and then the next year, for the ticks, they have available a lot of rodents to feed again. So they will survive the winter and increase and explode the population on year 2. And this is where the nymphs, which are the most important states for ticks, will bite the humans. Oops. Sorry. So we also gather a lot of data all over Switzerland to build a masting time series, and we will able to classify years depending if they have high masting, low masting, or intermediate masting, and no masting at all. So now we had 4 mechanistic pathways where we have data through the tick activity, through the forest ecology and the animals, and through the humans. We were missing one of the pathways, which actually is one of the remaining gaps that we have in this discipline. And then the approach was as following. So our idea is, let's build 2 models. Okay, the first model, the null model, is a model that is based only on seasonality and calendar patterns, and we will incorporate information, historical information from previous cases. So we will train the model on that. And those are very strong models. Actually, those are the models that we use for forecasting, because in reality, what happens this month is strongly correlated with what happened the month before and what happened that same month the previous year. But then we built our other model, which tried to show that this information around these three different mechanistic pathways was actually improving our predicting capacity. And indeed, our model was able to predict not just the total amount of cases of that year, but also the exact shape of the epidemic curve that we have. So I wanted to look at the data also around the children. So the problem with the children is that, as I said, we have a representation of 2% of the population, which is relatively low, and tick-borne encephalitis only includes the most serious cases. So we cannot look at children data by month, but we can aggregate them all together by year. And so what you see is the cases in Lyme in children under 16 on blue, the tick-borne encephalitis cases on red, and the masting effects. And what is cool is actually we can predict the excess of cases using masting in almost all the high years and all of the low levels. We were still missing 3 years, 2015, 2020, and 2022. So 2020, what we have is we didn't have masting 2 years before. So there was a lot of cases, but that was 2020, COVID-19. In Switzerland, a lot of children were going outside. The other 2 years, it's the opposite. We have a strong masting events, lagged 2 years, but we don't have a lot of cases. So actually, those 2 years have been among the top 3 years in terms of warming in Switzerland. And if you see at the time series carefully, you can see that in 2015 and in 2022, cases were rising up very fast in the spring, and then suddenly we have this sharp drop in humidity, which is critical for ticks, and then the epidemic curve just reduced. So, summarizing, predicting the risk of tick-borne diseases and vector-borne diseases in general is very complex. We have identified 4 dimensions: humans, habitats and animal hosts, vectors, and pathogens. For some of these dimensions, pathogens, we don't really know what is the influence of climate. And for some ecological process, such as masting, we don't know how climate in the long term will affect. We know that climate can have conflicting effects. So when we have milder temperatures in winters or in areas that were historically cold, warming will increase the risk of these diseases. But when we have heats and droughts, actually that reduces the risk. And finally, we have other processes such reforestation or the time we spend outdoors that somehow are affected by climate, but there are other forces that are independent from climate. So my 3 take-home messages are this. So first, complexity. I try to show you that understanding these processes, it's very complex, so we need to invest in understanding. And particularly for children, we have to change our conceptual mind towards life trajectories, because the vulnerabilities at early life and the cumulative effects that not only vector-borne diseases or infectious diseases have, but all the effects that climate change is on them, impacting on them. The second concept is that we have to rely on natural experiment, so we need to invest in good surveillance systems and good data systems, because this is the only tool that we really have to really answer what's going on. And finally, the last thing is acceleration. So climate change is not only going fast, it gets faster at every step. So what was true 10 years ago might not be true today. A disease that was rare here 5 years ago now is common. And as I said, we don't have the tools really to have always all the evidence. So we have to invest in preparing and not waiting until all the evidence is there, because we will just react. And we need also to assume that we will work always under a big uncertainty. Thank you very much.
Thank you very much. We've definitely learned some new things. And now it is my pleasure to welcome Professor Osterhaus, who will be speaking about One Health in a Warming World, preparing for zoonotic threats. Professor Osterhaus is a leading virologist. He's responsible for the discovery of many human and animal viruses. He's also a guest professor at the University of Veterinary Medicine, Hanover. But most importantly, we have inside information that today Professor Osterhaus is celebrating his birthday. So you have a warm congratulations. Warm congratulations from every ESPID members in this room. And please, your floor.
Well, thank you very much for this very nice introduction. And I'm very happy that you didn't mention my age. Yeah? So basically what I'm going to do is I'm going to, let me show the first slide. Okay. Okay, first of all, I'm very grateful for the invitation, obviously, for this fantastic meeting, and especially the title that was given to me by Susana Esposito, and One Health in a Warming World, Preparing for Zoonotic Threats. Well, it's quite a challenging title, and before embarking on that, you've heard the previous two presentations. It was great, and it showed to you how complex the situation is. We cannot just say climate change and it's going to change the world. So first, my affiliations and possible conflict of interest. I don't think I have a conflict of interest here. I think the most important thing for this paper is that I'm the co-chair of the Global One Health community and also editor-in-chief of the Nature Springer journal on that particular subject. Okay. Well, talking about One Health, we all know the concept, and I realize the audience is pediatricians, yeah? So perhaps a little bit of explanation. Yeah, One Health is really recognizes that the health of humans, animals, and the environment are closely connected. And during the talk, I will explain you what we have learned about that. So it's especially about the interfaces, and it's being claimed that almost 80% of the new human infectious disease, the emerging infections, are really coming from the animal world. And being a veterinarian by training, I've always been fascinated by viruses crossing the species barrier. Well, when we're going to talk about zoonosis, I think it's important to realize it's not only viruses. I'm a virologist, so we'll focus on viruses, but there is also bacterial zoonosis, parasitic, etc. We have heard a couple of examples earlier today. And so basically, I'm going to focus mainly on viruses. And so this is a very old slide I've used a lot for students, actually. And it's actually, we see emerging infectious diseases. And it illustrates, it's on the one hand, the microbial agents, be it viruses or bacteria, what have you, they have a capacity to adapt to a new situation. I'm not going to talk about that. The human host, if you realize how we have changed our habits over the past decades, if not century, I think it's enormous. I'm not going to talk about that too much, but also our environment. And when we talk about the human environment, it's the climate and the weather. We have had very nice examples there, and it has actually shown also that it's quite a complex situation. And perhaps on the next slide, which you see here, climate directly has an impact on human health through many factors. And you see that here. It starts with climatic extremes. We all know that air quality, sea level rise, which is a major thing. We saw an example there as well. Food production and water resources and infectious diseases. So it's a multi-headed monster, basically. And all these factors, they are interrelated as well. So I make it very complicated, I realize that. I'll give you some practical examples later on. And there's many additional factors, as you see here, Yeah, you see mobility of people, control measures, all these things, yeah, they affect the spread of viruses and the infections in humans as well. So this is from a nice paper in Nature Climate Change, where you see it's viruses, it's bacteria, it's animals, fungi, and go on. So it's not a simple thing. And I'm only going to talk about viruses. And so you realize that there's much more than that. But basically, it gives some very nice or bad examples. And when you look here on global warming, you have precipitation, floods, droughts, storms, all the way. There's so many things. And I'm mainly going to focus on vector-borne diseases. And you can directly criticize me. There's waterborne, there's airborne, there's direct contact, and all these other things are there. So the title I was given is really, well, limits me a little bit to what I can do. Now, so what I'm not going to talk about is climate change and antimicrobial resistance, but I wanted to mention it specifically because AMR is a major problem today. We all know that. Yeah, and in this particular paper, it's being illustrated at what different levels AMR is creeping into our equation, so to say. So I'm not going to talk about it any further because it would be a complete lecture. Okay, then and what we see here, basically this paper says that over half of the human pathogenic diseases can be aggravated by climate change. And at the top here, you see diseases influenced by climatic hazards. Yeah, and you see the different areas here. The blue area is really diseases exclusively aggravated. Then diseases here in the light blue area that are both aggravated and diminished. It could well be that certain diseases, when, for instance, the temperature goes up, you have less impact of that particular disease, and certain diseases might even very negatively be influenced. And then at the bottom here, when we look at infectious diseases here, you see again this separation here, and then you have diseases reported by the Global Infectious Disease and Epidemiology Network of CDC. It's a well-known thing. And then you see here diseases in the red block here. That's diseases not reported in this system. Then here, diseases reported here. And here, these diseases being aggravated. So it's a complex thing, and it's just an overview. And I'm going to focus more on a couple of details here. The IPCC assessment report already from 23 looked at climate and risk for human health due to some infectious diseases. And we have heard about Lyme disease today. Yeah, we have heard about dengue, and we heard about West Nile virus. And if you just look at the global warming here, and the expectation now is that we are going up about 2 degrees if we stay at the same level. But if we go up further, look at the risk here. With limited adaptation, the risk is going up dramatically. And you see here very high, moderate, and no influence. And we are already here in this reddish area for these 3 diseases. So just imagine if we are not limiting the global warming, we are really going to face a difficult situation in the future. I think this is one of the most impressive slides to me. Good. Well, when we look at the dynamics of arboviruses, and you've heard a lot about them already, I think it's quite important to see there's all these different things. We are talking mainly about climate change, but there's urbanization, there is global travel, and some of these spill over from wildlife. We heard about the Aedes aegypti, the albopictus, etc., and then the mitigating factors. Well, if there is a global expansion, as we have seen, these viruses, they tend to genetically adapt. Yeah, surveillance and diagnostics are important. Vector control challenges, I'll talk about that a little bit. And then we have emerging threats and public health implications. So it is a very complex situation there that we will try to manage. Well, you've seen a lot of maps already, and all these maps show that there is increasing prevalence. And here, this is about Aedes albopictus. Now, Aedes albopictus is a bad guy. It's a vector mechanism. It acts as a vector mechanism for many things. The red area that you see, here, that's where it originally comes from. The orange area is where we have seen it in the last 30 years. So it has made an enormous expansion also into Europe, which is quite important here. Yeah, and the yellow part is where we see it incidentally being introduced by ports, yeah, in harbors. So basically, well, this is one of the things I don't understand. When we have the situation of West Nile, around the turn of the century, West Nile virus was not known for North America or for the Americas at all. And then suddenly we got cases here. It started in Central Park, 1999, 2000. Mosquitoes introduced, yeah, probably introduced by aeroplanes from the Middle East. And then what we saw happening in 5 years' time, you see here the human cases are the blue ones, and the red ones, and the non-human cases are the blue ones. You see, this is really what we call a virgin soil epidemic. And we have seen in the previous talks that West Nile virus is already there in Europe. Why don't we see in 5 years this enormous shift there? It's going gradually over Europe, so we are missing. It is not just climate alone. There's much more than that. We don't understand the full situation. I have to mention Zika here as well. I'm not claiming this is climate related, but you see how viruses can circulate around the world. We all know the story of the microcephaly. Yeah, so this was a very bad story that is still going on. And then we have a similar thing about chikungunya virus. It was mentioned as well. They are out of Africa. Yeah, so basically these viruses, if there is a niche there, also stimulated by climate, it's going around very fast. We just heard about Tick-Borne encephalitis virus. It's an interesting story. When I started to work in Germany, I wrote a project for funding, and then I got a project to study the ecology of tick-borne encephalitis and the ticks, these different tick species. And you see here, the Netherlands was negative. So I had a similar project, but they wouldn't give it to me. And the year after that, we had the first outbreaks in the Netherlands. I'm not saying it's the climate, but we know that it's really the ticks, the area where the ticks occur are the major determinant. It's the Ixodes ricinus on the left-hand side, and it's other ticks on the right-hand side that actually spreading the virus. Well, this is from one of the papers by Susanne's group here, where she had a very nice overview. Different diseases caused by different pathogens. There's leishmaniasis here. There's still no malaria. Yeah, so it's very limited, of course, and there's a couple of other things. But interestingly, when you see the temperature raise, yeah, vector range expansion, yes. Then you see incubation period change, yes. Decreasing, obviously, and we see it's all pointing in the same direction. And then to make it a little bit more complicated, again, dengue, chikungunya, Zika, malaria here, yes, leishmaniasis, West Nile virus. So basically, when you look at this table, it's all humans here. Leishmaniasis, obviously, the sandflies also in dogs. And this is birds. It's quite interesting. It's a different mosquito group here. Then the spread. Yeah, you see it spreading into Europe. And then here you see this is into Europe. And you see everywhere is temperature rise, which is quite important, I think. Well, when we then look at the worldwide situation of dengue virus, and this is per 100,000. So Europe is not shown because the figures are too low, obviously. Yeah, but you see here basically in 3 months, at the beginning of this year, the end of last year, you see the situation here where how much it occurs in all over the world. And when we are then moving on, and then there is we see maps all the time. You have the suitability for Aedes albopictus, and you see that here. Yeah, you see the color codes here when it's dark red. Yeah, basically it's Spain, Italy, some areas here, and then and when you look here, epidemic risk of dengue, and you see it here, but also some areas. So you see that when it lights up more, that coincides quite well with the Aedes albopictus, and you see here some of these circles actually starting with autochthonous cases, cases really originating from infections that were acquired in the country itself. So it's moving on, as we have seen. And then the Aedes albopictus here, the suitability of Europe here, the density and the transmission risk. And if you look at the periods here, it's 2006 to 14, and then from 15 onward, and you see, here you see the difference being depicted, but you see it's moving on, so it's creeping up. So basically the suitability is creeping up due to the climate. the density follows. Yeah? You see that going up, and also the transmission risk. And again, we have seen that there are cases of dengue, and this is another interesting point here. We see clusters. Well, we see clusters of dengue virus, but here you see basically distribution of imported cases by month, symptoms, etc. You see the different years, 2019, 2021, etc. You see them going up. Here in 23 is the highest here, and then you look here in Paris. This is the Paris area here. Yeah, so Île-de-France, and you see here the more red it gets, the more suitable the situation becomes for the Aedes albopictus, and actually also the mosquitoes being there. And this actually resulted in 2023 for the first interfamiliar cluster of 3 autochthonous cases of dengue. These were the first one completely, well, not completely unexpected, but it's knocking on the door for sure. And so when we look at that, the tendency, 2005, 11, 18, and 24, and you see basically these are predictions here, but followed by actual incidences, observed colonization of Aedes albopictus. So it's creeping into Europe from the Mediterranean. And then when you look here, and we saw already some of these examples, so autochthonous cases of chick, dengue, and West Nile, the red one, the red area, there we have all 3 in the meantime. Yeah, here this is West Nile. And still, I'm a little bit puzzled why it's not sweeping over like it did in the US. So it's more complicated than just temperature, apparently. And then we have the dengue virus mainly here. There's more spots here and there now in 26 as well. But it's really in Portugal is the main region. So the general picture is the same all the time. We see all these infections coming on. So this is European CDC with a warning: Paris, Vienna, Zagreb, and other European cities will be at more risk of dengue, Zika, and chikungunya outbreaks as climate change expands mosquito range. So basically, we are to expect these kind of outbreaks, not only in Paris, but we are going to see them elsewhere, because the infections are being introduced by people all the time. When the mosquito is there, yeah, and there's a lot of other conditions that we heard in the previous talks, but basically, yeah, we are looking into that. And the other, you remember this IPCC prediction of what's going to happen. Yeah, with the very red, dark red areas, bars that you saw. So if we don't do anything, so we will be in trouble. So basically, what can we do? Well, basically, here is still an overview of the most important mosquito-borne diseases I discussed, native species to a limited extent, but the Aedes albopictus and also other invasive Aedes species are coming. We have here dengue, chikungunya, West Nile virus, and Zika virus. Those are the main ones, I would say. Well, there's insecticide resistance is mounting. Yeah, so we try. This is the climate thing, but here we have insecticides, but the insects getting resistant there. And when we go on, this is, I think, a nice overview here. So surveillance and control of mosquito-borne diseases in Mediterranean Europe. That's what we have to do. I'm not talking about vaccination at all, because vaccines are being developed, of course. And the major thing, in my view, the major thing to consider, is indeed to stop the global warming. Yeah? But that is, we all know that's a very hard nut to crack. But these are the things that can be done. Surveillance is important to know what's going on. Control of mosquito-borne diseases in Mediterranean Europe. Well, we have the source reduction. We can trap them. We can do biological control. Yeah, chemical treatments, as we have seen. Of course, there's regulations there. Well, there is some light on the horizon, I think. Yeah, so we can do genetic control. So we can have genetically modified mosquitoes that are not reproducing, especially the males that are not fertile anymore. We can even irradiate them. But I think this is a fantastic development here. Wolbachia, it's a natural thing it's a bacterium that infects the mosquitoes. And after being infected, it competes basically for the system for virus production. So the virus production in Wolbachia infected mosquito goes down tremendously. Yeah? So basically, this is technology that has been worked out quite in quite some countries in the meantime, it's being tried. And then we have, of course, smart technology and community involvement. We have to be aware, and the people have to know, what are the predisposing factors for these mosquitoes to breed, obviously. Well, just here, in short, male sterilization, either by irradiation, so irradiate males so that they cannot reproduce any longer. You can genetically modify them, you can combine these things, and you have the Wolbachia thing I mentioned, and it's really release that by aeroplanes, all kinds of different ways, so to really suppress the population. Well, again, if you look at the overview here, so we have extreme environmental changes. And obviously, if we want to do something, we have to block it here, go to the source. That's the most important thing. And that is very, very difficult. We have economic changes. We have demographic changes here. And so I think epidemiological surveillance of the diseases is quite important, but also entomological surveillance, and then really reduce, with the methods I showed you at the previous slide, try to reduce the fertility, at least, of the mosquitoes, and try to reduce them in that way. And as I said, there are quite some good examples of that. So I realize I focused very heavily on arboviruses because I think it's one of the major threats. But as I showed you in some of the previous slides, it's much more complicated than that. So tackling at the source, these extreme environmental changes, try in some way to reduce that is the most important thing, I think. And all the other things is monitoring what's happening. And then, of course, I haven't talked about vaccination against these infections. We have heard about TBE. We have a vaccine there. For dengue, there are vaccines. And vaccines for chikungunya and other things are emerging as well. So I think on the one hand, it's a very bleak situation. So basically, the climate change is a major thing. And especially for pediatricians, your patients are the ones going to suffer the most in the future. You should realize that. So basically, let's do something about it now. Thank you very much.
Thank you very much, Professor Osterhaus. And we welcome questions from the audience. You can, of course, approach the floor microphones. And for our off-site participants, you can send your questions through the meeting app. And until we have questions from the audience, I'd like to ask the three of you maybe, which vector-borne disease are you most concerned about, especially for children in Europe?
Worldwide or here in Europe?
You can answer both.
Please use the mic.
Sorry.
It's an impossible question.
Yeah, it's a very difficult question. I mean, I think regarding tick-borne diseases, at least, clearly, Lyme borreliosis and tick-borne encephalitis are very important from the public health perspective because of different reasons. I would say you have also Crimean Congo hemorrhagic fever, which is quite scaring. We already have some endemic cases in Spain, Portugal, Greece. And this is, we don't know a lot about it. So, and the mortality rate in adults is quite high, and children, it's still contested. I think only people in Turkey, I think they have some series in Turkey. But nevertheless, I think, depending on how you look at it, but those will be my pick for ticks.
So I fully agree, but just to add, of course, worldwide, malaria is one of the key things. Vaccines are forthcoming at the moment. So that's malaria, but IPCC 2023 really singled out in their predictions the flavivirus on the one hand and the Lyme disease on the other hand. So, and I think that is for a reason. So I think that might be the answer to your question.
Okay.
So Pablo in the audience, please.
Pablo Rojo, Madrid, Spain. Fantastic speakers. Thank you very much. My question is for Pablo, related to the different aspects you evaluated, and it's also related to all speakers. And you presented very nicely the different other considerations we have to take into. But I was wondering about population, increase of population. Have you analyzed that or included that in your models?
Yes, I mean, yes, this is. I mean, we won't expect to the Swiss population to change a lot in the past 20 years. Actually, we compute its incidence, so a way to adjust. But that's true. I mean, actually, for malaria, this is something that I always think, because in the past 10 years we see a stallment in the number of cases, but on the other hand, African population, which basically is like 90% of the malaria cases, has exploded too. So that's a very relevant point. Yeah.
But you also showed very nice pictures about the population of the intermediate host. So the rodents and the deer on the other hand. So there's fluctuations there. So human population is important, but I think in looking at voles, looking at deer is quite an important thing to do as well.
Thank you.
So if there's nothing from the audience at the moment, so Romeo, what would you say, because you said for you it's the first time in such a meeting. I think it's also for ESPID one of the first bigger sessions that we do on that topic actually. What would be your wish for the collaboration with us as pediatric infectious disease persons to what would we need to foster to bring that topic more forward in the future?
Yeah, good question. I think we are, in my opinion, we are on the good way. The One Health approach, strategy is becoming more and more popular in different disciplines. I say that as a vector ecology, we used to do One Health without knowing the One Health paradigm many years ago, because in our field, vector ecology was the way to see the complexity of the thing together and the interrelation between environmental activity, climatic activity, human activity, animal activity. So I don't see mistake in our approach by now. I see that collaboration is becoming natural because we always understand more and more the complexity and the interrelation between things, and we know that we need each other. So I think that I am very positive on this.
And you mentioned that that it's quite difficult actually sometimes to decipher between what is actually the climate change and what is actually the natural development. So what do you think is there the future actually to the tools? I mean, you talked about modeling, so and maybe AI. So what are the strategies to tackle this decipher process? What is the hen? What is the egg?
I gave one example where I don't understand why West Nile virus is not sweeping over Europe. Yeah, because the situation is quite similar and the climatological condition is quite similar as in North America. And there it started in Central Park, and in 2-3 years it swept all over the country. Yeah, and so what is different? And I think it's a very difficult question to answer. But I think, as we were trying to do today, yeah, all the three speakers, is really delineate what are the major factors there. What are the hosts? What are the intermediate hosts? What is temperature? What is rain? What is floodings? So it's all these things together. And I think the predictions there are very difficult. And I think, fortunately, at the end of the day, we also have intervention strategies with humans. Of course, I've been focusing mainly on the vectors, you know, and I think it's fantastic what's happening there, especially the story with the bacteria, with the Wolbachia, etc. We have to roll that out, obviously, but at the end of the day, in humans, we will have intervention strategy, because Tick-Borne encephalitis and the other flavivirus, but also chikungunya virus related diseases are devastating with high incidences, high mortality even. And so I think it's important that we have vaccines and antivirals, effective antivirals and vaccines. And I should say the vaccines that we have today need to be improved even, you know. And so fortunately we have a number of vaccines now against the flavivirus, but I think that's the way to go. But the most important thing is to stop the global warming, of course, but that's a point that we cannot address here, I'm afraid.
Please.
And also introduce yourself, where you're from.
Sure.
And, yeah.
Fantastic lectures. Thank you very much. I'm Masaki Yamada from Tokyo, Japan, National Children's. The climate changes seems to have a lot of impact on the vector ecology, but I'm interested in how these changes can potentially impact on the pathogen factors directly. Can you comment on that?
Sorry, what?
So could you repeat the last part of your question?
The pathogen itself, virus or maybe the pathogen. Not, so you guys spoke a lot about the vector ecology standpoint, but would that be any impact the climate changes or environmental changes directly changes the pathogen, the features of the pathogen or pathogenicity of that?
So you mean the interaction between climate change, direct interaction with climate change and pathogens themselves?
Yeah.
That's what you mean?
Mm-hmm.
Well, that's a difficult question to answer, but we see the pathogens, by definition, have an enormous capability of adaptation. So they mutate, etc. And in one of the slides I showed that you see immediately that if the climate gets warmer, yeah, then just 1 or 2 degrees, you see the replication cycle, it goes much faster. So I think that is one part of the whole equation, I think. Yeah.
Yeah, I will say that as I try to explain, this is a major gap that we have. And indeed, I would like to say that, for example, for malaria, and this is very, very difficult to measure because you can measure it in labs and experiments, but this is not necessarily real conditions. So for malaria, I work a lot in malaria, and malaria has relied a lot on mathematical models, as my colleague presented. One variable that we still are very unsure is how temperature influences the replication of the malaria parasite in the mosquito. And when you take the equation that governs this type of models, that factor is extremely important, but nobody knows really if this is true or not. It happens in laboratories, but we cannot just have mosquitoes, put them parasites, send them outside and wait and then pick them back. And so it's a very hard problem, but definitely we need more people with clever people to try to figure out what are the...
Well, one of the things that has not been mentioned yet, when we talk about vaccination and intervention strategies, I think one of the lines of research at the moment is also how to deal with vaccination with components of saliva of the ticks, for instance, or the products that the mosquitoes use to get to suck the blood, and there you have possibilities as well. So and what you said, we need smart scientists that really, from the entomological world as well, to try and look at the whole problem from different angles. If that's an answer to your question, yeah. Okay.
Yeah, I'd like to bring an example, which is the correlation between Aedes albopictus, which is a species which is spreading around the world, and the chikungunya. What happened is that by the introduction of albopictus in some part of the world where there is more capacity of the pathogen of the chikungunya virus to survive, the pathogen develop through a mutation a strain which is much more efficient in being transmitted by albopictus. So there is a coevolution between the vector and the pathogen due to the fact that before they were never being in contact or much less in contact. With the new contact, the capacity of the pathogen to develop new strain to be able to so there is the evidence already that the spread of the vector and the interaction with new pathogen may bring the system to be more efficient in terms of the epidemiological impact.
Last question on the floor.
Hello, yes. Thank you for the excellent talks, very much. Norman Lappin, University of Texas Southwestern Medical Center. I just very curious how to address the question of teasing out pediatric data, especially, we're studying necrotizing fasciitis, for example, and there's just differences in mortality, and such incidents from adult and children, but then there's kind of a slope. I mean, where do you hit the cutoff? And also the fact that the cases are so rare to begin with. I know it's a different topic, but just in general, teasing out children is, I think, pretty challenging, from the general population data. So just curious how you would go about addressing that from a data analysis, extraction and analysis, maybe multi-study perspective.
That's a tough one. I will say try different approaches. That's the one thing that I've learned with modeling is that actually you can try several things at the same time and look for what is what is the middle point. I don't know. Sorry.
Okay. Well, thank you.
I think, if I may add, I think children are also humans, and the only difference there that we see is they usually are born with an immature immune system, and so at the very early age they may be much more susceptible. That's also what we tend to see. But at a later age, then basically, if they're fully immunocompetent, so they behave like in general like normal people. There's another issue, of course, we didn't talk about it at all. But if you talk about the flaviviruses, so immunity to one virus, for instance, Zika virus, and you get later infected similar to the different strains of dengue, you get later infected by another flavivirus, you may get an enhanced disease as well. And and so this dengue shock syndrome, for instance, is something we see in children quite frequently.
I think it can also reverse because of this concept of resilience in children compared to some older adults. I mean, I guess it depends on the disease.
Okay, we want to thank the audience for their questions and, of course, thank our three speakers for a fascinating session. We see that ID is evolving.
Yeah, we close the session, and I think this topic showed that there's room for more, and we'll catch up with this in 2027 in Berlin, I would say. And then we have now a short break and see each other in that room in 15 minutes again for the next session. Stay tuned, please.